What if the thing keeping you alive right now is also the thing you understand least about yourself? Most people know their blood type the way they know their shoe size. A number, a letter, a fact filed away and rarely examined. O positive, O negative, one of those. You probably learned it once and moved on. But the difference between those two types, the line that separates them, is not what most people think it is. For decades, medicine described it as a single protein, present or absent, simple, manageable, a detail that mattered in emergencies and almost nowhere else. That description was not wrong, it was just nowhere near complete. The Rh factor, the protein that makes blood positive or negative, turns out to be the surface of something far deeper. A difference that reaches back hundreds of thousands of years, that shapes how your immune system responds to threat, how your body handles pregnancy, how emergency rooms make life or death decisions in under 60 seconds. The gap between O positive and O negative was never really about one protein. It was about what that protein represents. And what it represents is stranger, older, and more consequential than anyone who glances at a blood type card is ever told. There is a protein that sits on the surface of your red blood cells. It is called the Rh factor, and whether you have it or you do not have it is the entire difference between testing positive or negative for your blood type. That is the official explanation. It is accurate as far as it goes. It does not go very far. The Rh factor is not a single molecule. It is a complex of proteins encoded by two closely related genes, RhD and RhCe, sitting on chromosome 1. The RhD gene is the one that matters for the positive and negative distinction. If it is active and producing its protein, you are Rh positive. If it is absent or silenced, you are Rh negative. O positive and O negative blood share everything else. Neither carries the A antigen, neither carries the B antigen. In every other structural sense, they are the same blood moving through the same vessels doing the same work. The single presence or absence of that RhD protein is what separates them. For most of the 20th century, medicine treated this as a manageable administrative distinction. Type your patient, match the blood, avoid the mismatch, move on. The Rh factor appeared on intake forms and hospital wristbands as a footnote to the more important ABO classification. It was considered the smaller variable. What subsequent decades of research revealed is that this framing had the emphasis exactly backwards. The Rh protein complex is one of the most structurally significant components of the red blood cell membrane. It is not decorating the surface of your cells. It is embedded in it, playing a role in cellular integrity that researchers are still mapping. Its complete absence does not simply remove a protein. It alters the architecture of the cell itself in ways that are measurable, reproducible, and not yet fully understood. The distinction between O positive and O negative begins with one protein. It does not end there. 7%. That is the size of the O negative donor pool. Not 7% of willing donors, 7% of the entire human population. In a city of 1 million people, 70,000 individuals carry the only blood type that can be transfused into any patient regardless of their own type, regardless of whether there is time to test them, regardless of anything except whether the supply is available. In a mass casualty event, that supply runs out fast. A single trauma patient with severe internal bleeding can require dozens of units before the bleeding is controlled. The arithmetic is unforgiving. The demand is not theoretical. It happens every day in emergency rooms that are one bad night away from calling other hospitals asking if anyone has units to spare. O negative earned the title universal donor because it carries none of the surface antigens that trigger immune rejection. No A antigen, no B antigen, no Rh protein. There is nothing on its surface for a recipient's immune system to identify as foreign. It can enter any bloodstream without triggering the attack response that makes mismatched transfusions fatal. In the 60 seconds before a trauma patient loses consciousness, that compatibility is the difference between a decision and a gamble. But the universal donor label obscures something important. O-negative donors are the most restricted receivers in the entire blood type system. They can give to everyone. They can only receive from their own kind. If an O-negative patient arrives unconscious and bleeding, and the hospital's O-negative supply is exhausted, O-positive blood sitting 3 feet away in the same refrigerator cannot be used. It would trigger an immune response that compounds the original injury. The universality runs in one direction only. O-positive blood tells a different story. Carried by 38% of the population, it is the most transfused blood type on Earth. The majority of people can receive it safely. It is the workhorse of emergency medicine, the type that fills the gap when nothing else is available. But it cannot fill that gap for everyone. The portion of the population that is Rh negative cannot receive it without risk. The most common blood type on Earth is still the wrong blood for millions of people. When the wrong blood enters your body, your immune system does not hesitate. It does not wait for confirmation or run a secondary check. It identifies the foreign protein, classifies it as a threat, and launches a response designed to destroy it. In the context of a mismatched transfusion, that response is called a hemolytic reaction, and it is exactly as serious as the name implies. The transfused cells are attacked and broken apart. Hemoglobin spills into the bloodstream where it does not belong. The kidneys, tasked with filtering the sudden toxic load, can fail. Blood pressure drops. The body goes into shock. What was meant to save the patient accelerates the crisis instead. This is not a rare edge case in transfusion medicine. It is the central risk that the entire blood typing system exists to prevent. The Rh factor sits at the middle of it. An Rh negative patient who receives Rh positive blood may not react catastrophically on the first exposure. The immune system takes time to build antibodies, but it remembers. A second exposure triggers a faster and far more aggressive response. The body has already cataloged the foreign protein and prepared for it. The reaction on second contact can be severe enough to be fatal. Nowhere is this immunological memory more consequential than in pregnancy. When an Rh negative mother carries an Rh positive baby, a small amount of fetal blood can cross into the maternal circulation during delivery. The mother's immune system encounters the Rh protein for the first time and begins producing antibodies against it. The first pregnancy often proceeds without incident. The second one does not. In a subsequent pregnancy with another Rh positive baby, those maternal antibodies cross the placenta. They enter the fetal bloodstream and begin attacking the baby's red blood cells from inside the womb. The resulting condition, hemolytic disease of the newborn, can cause severe anemia, organ damage, and death before the child is born. The immune system is not malfunctioning. It is doing precisely what it was built to do. It encountered something it did not recognize, it built a defense, and it deployed that defense at the next opportunity. The tragedy is that the thing it identified as foreign was its own child. Type O blood is old. Not old in the way that feels like a footnote to human history. Old in the way that reframes what human history actually is. Genetic evidence suggests type O is the oldest of the ABO blood types, likely predating the others by hundreds of thousands of years, possibly longer, making it almost certainly the ancestral blood type from which A, B, and AB emerged later as adaptations. The leading hypothesis among evolutionary biologists is that mutations in the ABO gene altered the surface of red blood cells in response to environmental pressures, to pathogens, to the specific disease landscapes of different regions. Type O persisted because it worked. It offered its own set of immunological advantages that kept it dominant across populations even as new variants appeared. Today, it remains the most common blood type on Earth, carried by roughly 44% of the global population. But the distribution of O negative within that broader O category is not uniform, and the pattern of that distribution has quietly occupied researchers for decades. O negative appears at its highest frequencies in populations of Basque origin in northern Spain and southwestern France, where some studies have recorded prevalences among the highest in the world, more than 4 times the global average. It drops sharply, moving east across Europe, falls further across Africa and Asia, and reaches some of its lowest recorded frequencies in East Asian populations. That gradient does not follow the patterns of other genetic markers in any straightforward way. It does not map cleanly onto migration routes or founder effects that explain similar distributions elsewhere. What it suggests is a specific population history that researchers have not yet fully reconstructed. One thing before the final piece. Everything I just showed you about O-negative blood and its ancient distribution is traceable inside your own DNA results. I wrote the guide for exactly that, It is called Where Do You Really Come From? Link is below. Now, here is what nobody talks about. The RHD gene does not mutate into the Rh negative state. That distinction matters more than it might initially appear. A mutation implies a change, an alteration of existing genetic material producing a different outcome. What happens in Rh negative individuals is something structurally different. The RHD gene is absent. In most people with Rh negative blood, the gene that would produce the Rh protein has been deleted from chromosome 1 entirely. The sequence that should be there is not there. There is no corrupted version, no silenced copy, no switched off instruction. The instruction does not exist. Geneticists describe this as a gene deletion, and it is common enough in population genetics that it does not by itself imply anything exotic. Gene deletions happen. They accumulate across evolutionary time, and some of them persist because they are neutral or because they confer advantages that outweigh the costs. The RHD deletion falls into a category of structural variants that researchers are still characterizing. What makes it notable is not its existence, but what happens when scientists try to reverse it. In laboratory settings, researchers have attempted to introduce functional RHD sequences into Rh negative blood cells using gene editing tools. The goal was straightforward. If the absence of the gene is the problem, supply the gene and solve the problem. The results were not straightforward. Cells that successfully incorporated the RHD sequence frequently became unstable. Some failed to produce functional protein. Others showed disrupted membrane architecture. The red blood cell, it turns out, does not simply accommodate the addition of a protein whose structural role it has never been built around. The absence of the RHD gene in Rh negative individuals appears to have downstream effects on how the cell membrane is organized at a fundamental level. Removing the protein did not leave a gap. It changed the blueprint. This is not evidence of extraterrestrial origin or parallel evolution, claims that circulate widely and rest on nothing. It is evidence that the relationship between a single gene and the broader architecture of a cell is more complex and more integrated than the original model of a simple protein switch ever accounted for. Type O blood has spent hundreds of thousands of years in contact with some of the worst pathogens in human history, and the relationship is written into its biology in ways that are only now becoming clear. The resistance profile of type O blood reads like a record of every epidemic that ever threatened to end the human experiment. Malaria, one of the oldest and most persistent killers in human evolutionary history, is significantly less able to cause severe disease in type O individuals. The mechanism involves a protein called rifin, which the malaria parasite uses to clump red blood cells together, blocking vessels and causing the organ damage that kills. Type O cells bind rifin far less efficiently than A or B cells. The clumping does not happen at the same rate. The severe outcomes are measurably rarer. Cholera tells a similar story from a different angle. Type O individuals show dramatically higher rates of severe cholera when infected, not resistance but vulnerability, suggesting that whatever selective pressure shaped the type O immune profile was not optimizing against every pathogen equally. It was making trade-offs. That trade-off logic runs through the entire disease susceptibility profile of type O blood. Lower rates of cardiovascular disease, reduced risk of certain clotting disorders, better outcomes in some viral infections, including documented differences in COVID-19 severity across multiple studies. Against those advantages sits increased susceptibility to certain bacterial infections, ulcers driven by H. pylori, and the severe E. coli strain O157, where studies have found type O individuals disproportionately represented among the most serious cases. What this profile describes is not a blood type that is better or worse than others. It is a blood type that is specialized, shaped by hundreds of thousands of years of specific environmental pressures into something optimized for a particular set of threats at the cost of increased exposure to others. The biology is not random. It is the accumulated record of every disease that ever forced a population to adapt or disappear. Type O survived. The terms of that survival were never free. The assumption that guided transfusion medicine for most of the 20th century was that the Rh factor was a binary footnote, present or absent. Match it, avoid the mismatch, move on. That assumption was not irrational given what was known at the time. It was a working model built from the tools available. What the last 2 decades of molecular genetics have done is reveal how much the working model left unexamined. The Rh protein complex is now understood to be one of the most abundant, non-glycosylated proteins in the red blood cell membrane. It is not sitting on the surface as a passive marker. It is structurally integrated into the membrane in a way that influences the cell's shape, its flexibility, its ability to move through narrow capillaries without rupturing. Cells that lack it entirely, as in Rh-negative individuals, compensate through alternative membrane arrangements that researchers are still mapping. The clinical implications of that compensation are not yet fully characterized. What has been overturned more dramatically is the popular narrative surrounding Rh-negative blood, the one that attributes its origins to alien intervention, ancient genetic engineering, or a separate evolutionary lineage. That narrative is not a fringe curiosity. It circulates widely, generates significant engagement, and is believed with genuine conviction by a substantial number of people. It is also not supported by any credible evidence. The RHD deletion is a structural variant with a traceable evolutionary history. The clustering of Rh-negative blood in Basque populations reflects specific population genetics, not extraterrestrial ancestry. The physical traits attributed to Rh-negative individuals, the higher IQs, the lower body temperatures, the paranormal experiences, have not survived any rigorous investigation. What the science has actually overturned is quieter than alien intervention and considerably more interesting. It overturned the idea that a single protein difference is a simple thing. It revealed that one missing gene reshapes cell architecture, influences disease susceptibility across millennia, determines who can save whom in an emergency room, and connects living people to an evolutionary history stretching back further than most people ever consider. The protein was always the beginning of the story. It was never the whole thing. Your blood type is not a footnote. It is not an administrative detail filed away on a hospital wristband and forgotten until the next emergency. It is a biological record that connects you to decisions made by evolution long before you existed, to pathogens that shaped entire civilizations, to a single gene deletion whose downstream consequences researchers are still tracing at the level of individual cell membranes. The difference between O-positive and O-negative was described for decades as a protein. One present, one absent. Clean, simple, closed. That description was the beginning of an answer, mistaken for the whole answer. What the science has since revealed is a difference that reaches into cellular architecture, into ancient disease history, into the immunological logic of pregnancy, into the emergency room calculus that decides in under a minute which blood can enter which body without triggering a war inside it. None of that was visible from the surface of a protein. The research is not finished. The RHD deletion and its structural consequences are still being mapped. The evolutionary history of O-negative's geographic distribution has not been fully reconstructed. The disease susceptibility profiles are still generating new findings. Science rarely produces a clean final answer on questions this old and this layered. What it produces instead is a picture that keeps getting more detailed, more precise, and more surprising the longer anyone looks at it. The protein was never the whole story. It was the first visible edge of something far older, far more structurally significant, and far more consequential than anyone understood when they first gave it a name.